I think we tried to bring this question up a couple of times, but let's return to it again. Do you think is the difference between the way a computer represents information, the human genome represents and stores information? Like what, and maybe broadly, what is the difference between how you think about computers and how you think about biological systems?
So I made a very provocative claim earlier that we are a digital computer, like that at the core lies a digital code. And that's true in many ways, but surrounding that digital core, there's a huge amount of analog. If you look at our brain, it's not really digital. If you look at our sort of RNA and all of that stuff inside our cell, it's not really digital. It's really analog in many ways. But let's start with the code and then we'll expand to the rest. So the code itself is digital. So there's genes. You can think of the genes as, I don't know, the procedures, the functions inside your language. And then somehow you have to turn these functions on. How do you call a gene? How do you call that function? The way that you would do it in old programming languages is go to address whatever in your memory, and then you'd start running from there. And, modern programming languages have encapsulated this into functions and objects and all of that. And it's nice and cute, but in the end, deep down, there's still an assembly code that says go to that instruction, and it runs that instruction. If you look at the human genome and, the genome of pretty much most species out there, there's no go-to function. You just don't start in, transcribing in position 13,527 in chromosome 12. You instead have content-based indexing. So at every location in the genome, in front of the genes that need to be turned on, I don't know, when you drink coffee, there's a little coffee marker in front of all of them. And whenever your cells that metabolize coffee need to metabolize coffee, they basically see coffee and they're like, ooh, let's go turn on all the coffee marked genes. So there's basically these small motifs, these small sequences that we call regulatory motifs. They're like patterns of DNA. They're only 8 characters long or so, like GAT, GCA, etc. And these motifs work in combinations, and every one of them has some recruitment affinity for a different protein that will then come and bind it. And together, collections of these motifs create regions that we call regulatory regions that can be either promoters near the beginning of the gene, and that basically tells you where the function actually starts, where you call it, and then enhancers that are looping around on the DNA that basically bring the machinery that binds those enhancers and then bring it onto the promoter, which then recruits the right sort of the ribosome and the polymerase and all of that thing, which will first transcribe and then export and then eventually translate in the cytoplasm, whatever RNA molecule. So the beauty of the way that the digital computer, that's the genome, works is that it's extremely fault tolerant. If I took your hard drive and I messed with 20% of the letters in it, of the zeros and ones, and I flipped them, you'd be in trouble. If I take the genome and I flip 20% of the letters, you probably won't even notice. And that resilience,
That's fascinating, yeah,
is a key design principle. And again, I'm anthropomorphizing here, but it's a key driving principle of how biological systems work. They're first resilient and then anything else. And when you look at this incredible beauty of life from the most, beautiful, human genome, maybe, of humanity and all of the ideals that should come with it, to the most terrifying genome, like, COVID-19, SARS-CoV-2, and the current pandemic, you basically see this elegance as the epitome of clean design. But it's dirty. It's a mess. It's, the way to get there is hugely messy. And that's something that we as computer scientists don't embrace. We like to have clean code. As like in engineering, they teach you about compartmentalization, about sort of separating functions, about modularity, about hierarchical design. None of that applies in biology.
testing. Yeah.
Testing, sure. Yeah, biology does plenty of that. But through evolutionary exploration.
Yeah.
But if you look at biological systems, first, they are robust, and then they specialize to become anything else. And if you look at viruses, the reason why they're so elegant when you look at the design of this, genome, it seems so elegant. And the reason for that is that it's been stripped down from something much larger because of the pressure to keep it compact. So many compact genomes out there have ancestors that were much larger. You don't start small and become big. You go through a loop of add a bunch of stuff, increase complexity, and then, slim it down. And one of my early papers was, in fact, on genome duplication. One of the things we found is that baker's yeast, which is the, yeast that you use to make bread, but also the yeast that you use to make wine, which is basically the dominant species when you go in the fields of Tuscany and you say, what's out there, it's basically Saccharomyces Cerevisiae, or the way my Italian friends say, Saccharomyces Cerevisiae.
Which means what?
Saccharomyces. Okay, I'm sorry. I'm Greek. So yeah, Saccharomyces. Saccharomyces is sugar. Myces is fungus.
Yes.
Cerevisiae, cerveza, beer. So it means the sugar fungus of beer.
Yeah.
Less sounding to the ear.
Still poetic, yeah.
So anyway, Saccharomyces cerevisiae, basically the major baker's yeast out there, is the descendant of a whole genome duplication. Why would a whole genome duplication even happen? When it happened is coinciding with about 100 million years ago and the emergence of fruit-bearing plants. Why fruit-bearing plants? Because animals would eat the fruit, would walk around and poop huge amounts of nutrients along with a seed for the plants to spread. Before that, plants were not spreading through animals, they were spreading through wind and all kinds of other ways. But basically, the moment you have fruit-bearing plants, these plants are basically creating this abundance of sugar in the environment. So there's an evolutionary niche that gets created. And in that evolutionary niche, you basically have enough sugar that a whole genome duplication, which initially is a very messy event, allows you to then, relieve some of that complexity.
So I had to pause. What does genome duplication mean?
That basically means that instead of having 8 chromosomes, you can now have 16 chromosomes.
So, but the duplication at first, when you have when you go to 16, you're not using that.
Oh yeah, you are. Yeah. So basically, from one day to the next, you went from having 8 chromosomes to having 16 chromosomes. Probably a non-disjunction event during a division. So you basically divide the cell. Instead of half the genome going this way and half the genome going the other way after duplication of the genome, you basically have all of it going to one cell. And then there's sufficient messiness there that you end up with slight differences that make most of these chromosomes be actually preserved. It's a long story short to basically...
But it's a big upgrade, right? So that's...
Not necessarily, because what happens immediately thereafter is that you start massively losing tons of those duplicated genes. So 90% of those genes were actually lost very rapidly after whole genome duplication. And the reason for that is that biology is not intelligent. It's just ruthless selection, random mutation. So the ruthless selection basically means that as soon as one of the random mutations hit one gene, ruthless selection just kills off that gene. It's just, if you have a pressure to maintain a small compact genome, you will very rapidly lose the second copy of most of your genes. And a small number, 10%, were kept in 2 copies. And those had to do a lot with environment adaptation, with the speed of replication, with the speed of translation, and with sugar processing. So I'm making a long story short to basically say that evolution is messy. The only way, the example that I was giving of messing with 20% of your bits in your computer, totally bogus. Duplicating all your functions and just throwing them out there in the same, function, just totally bogus. Like this would never work in an engineer system. But biological systems, because of this content-based indexing and because of this modularity that comes from the fact that the gene is controlled by a series of tags, and now if you need this gene in another setting, you just add some more tags that will basically turn it on also in those settings. So this gene is now pressured to do 2 different functions, and it builds up complexity. I see whole genome duplication and gene duplication in general as a way to relieve that complexity. So you have this gradual buildup of complexity as functions get sort of added onto the existing genes, and then boom, you duplicate your workforce, and you now have two copies of this gene, one will probably specialize to do one, and the other one will specialize to do the other, or one will maintain the ancestral function, the other one will sort of be free to evolve and specialize while losing the ancestral functions, and so on and so forth. So that's how genomes evolve. They're just messy things, but they're extremely fault tolerant, and they're extremely able to deal with mutations because that's the very way that you generate new functions. So neofunctionalization comes from the very thing that breaks it. So even in the current pandemic, many people are asking me, which mutations matter the most? And what I tell them is, well, we can study the evolutionary dynamics of the current genome to then understand which mutations have previously happened or not, and which mutations happen in genes that evolve rapidly or not. And one of the things we found, for example, is that the genes that evolved rapidly in the past are still evolving rapidly now in the current pandemic. The genes that evolved slowly in the past are still evolving slowly.
Which means that they're useful?
Which means that they're under the same evolutionary pressures. But then the question is, what happens in specific mutations? So if you look at the D614 gene mutation that's been all over the news, so in position 614, in the amino acid 614 of the S protein, there's a D2G mutation that sort of has creeped over the population. That mutation, we found out through my work, disrupts a perfectly conserved nucleotide position that has never been changed in the history of millions of years of equivalent mammalian evolution of these viruses. That basically means that it's a completely new adaptation to human. And that mutation has now gone from 1% frequency to 90% frequency in almost all outbreaks.
So there's a mutation. I like how you said in the 416. What is okay?
Yeah, 614. Sorry,
614.
D614G.
D6. So literally, what you're saying is this is like a chess move.
Yeah.
So it just mutated one letter to another.
Exactly.
And that hasn't happened before.
Yeah.
And
never.
And this mutation is really useful.
It's really useful in the current environment of the genome, which is moving from human to human. When it was moving from bat to bat, it couldn't care less for that mutation. But it's environment specific. So now that it's moving from human to human, it's moving way better, like by orders of magnitude.
So what do you? Okay, so you're like tracking this evolutionary dynamics, which is fascinating. But what do you do with that? So what does that mean? What does this mean? What do you make? What do you make of this mutation in trying to anticipate? I guess is one of the things you're trying to do is anticipate how this unrolls into the future. This evolutionary dynamic.
It's such a great question. So there's two things. Remember when I was saying earlier, mutation is the path to new things, but also the path to break old things. So what we know is that this position was extremely preserved through gazillions of mutations. That mutation was never tolerated when it was moving from bats to bats. So that basically means that position is extremely important in the function of that protein. That's the first thing it tells. The second one is that position was very well suited to bat transmission, but now is not well suited to human transmission, so it got rid of it. And it now has a new version of that amino acid that basically makes it much easier to transmit from human to human. So in terms of the evolutionary history teaching us about the future, it basically tells us here's the regions that are currently mutating. Here's the regions that are most likely to mutate going forward. As you're building a vaccine, here's what you should be focusing on in terms of the most stable regions that are the least likely to mutate, or here's the newly evolved functions that are the most likely to be important because they've overcome this local maximum that it had reached in the bat transmission. So anyway, it's a tangent to basically say that evolution works in messy ways. And the thing that you would break is the thing that actually allows you to first go through a lull and then reaching new local maximum. And I often like to say that if engineers had basically designed evolution, we would still be perfectly replicating bacteria. Because it's by making the bacterium worse that you allow evolution to reach a new optimum.
just to pause on that, that's so profound. That's so profound for the entirety of this scientific and engineering disciplines.
Exactly. We as engineers need to embrace breaking things. We as engineers need to embrace robustness as the first principle beyond perfection, because nothing's going to ever be perfect. And when you're sending a satellite to Mars, when something goes wrong, it'll break down, as opposed to building systems that tolerate failure and are resilient to that, and in fact get better through that.
So the SpaceX approach versus NASA for the
for example.
Is there something we can learn about the incredible, take lessons from the incredible biological systems in their resilience, in the mushiness, the messiness, to our computing systems, to our computers?
It would basically be starting from scratch in many ways. It would basically be building new paradigms that don't try to get the right answer all the time, but try to get the right answer most of the time, or a lot of the time.
Do you see deep learning systems and the whole world of machine learning as kind of taking a step in that direction?
Absolutely. Absolutely. Basically, by allowing this much more natural evolution of these parameters, you basically, and if you look at sort of deep learning systems, again, they're not inspired by the genome aspect of biology. They're inspired by the brain aspect of biology. And again, I want you to pause for a second and realize the complexity of the entire human brain, with trillions of connections within our, neurons, with millions of cells talking to each other, is still encoded within that same genome. That same genome encodes every single freaking cell type of the entire body. Every single cell is encoded by the same code. And yet, specialization allows you to have the single viral-like genome that self-replicates, the single modular automaton, work with other copies of itself. It's mind-boggling. Create complex organs through which blood flows. And what is that blood? The same freaking genome. Create organs that communicate with each other. And what are these organs? The exact same genome. Create a brain that is innervated by massive amounts of blood pumping energy to it, 20% of our energetic needs, to the brain from the same genome. And all of the neuronal connections, all of the auxiliary cells, all of the immune cells, the astrocytes, the oligodendrocytes, the neurons, the excitatory, the inhibitory neurons, all of the different classes of pericytes, the blood-brain barrier, all of that, same genome.
One way to see that in a sad, this one is beautiful. The sad thing is thinking about the trillions of organisms that died to create that.
You mean on the evolutionary path to humans?
On the evolutionary path to humans. It's crazy. There's two descendant of apes just talking on a podcast. Okay. It's just so mind-boggling.
Just to boggle our minds a little bit more.
Yeah.
Us talking to each other. We are basically generating a series of vocal utterances through our pulsating of vocal cords received through this. The people who listen to this are taking a completely different path to that information transfer, yet through language. But imagine if we could connect these brains directly to each other. The amount of information that I'm condensing into a small number of words is a huge funnel, which then you receive and you expand into a huge number of thoughts from that small funnel. In many ways, engineers would love to have the whole information transfer. Just take the whole set of neurons and throw them away. Throw them to the other person. This might actually not be better, because in your misinterpretation of every word that I'm saying, you are creating new interpretation that might actually be way better than what I meant in the first place. The ambiguity of language perhaps might be the secret to creativity. Every single time you work on a project by yourself, you only bounce ideas with one person, and your neurons are basically fully cognizant of what these ideas are. But the moment you interact with another person, the misinterpretations that happen might be the most creative part of the process. With my students, every time we have a research meeting, I very often pause and say, let me repeat what you just said in a different way. And I sort of go on and brainstorm with what they were saying, but by the third time, it's not what they were saying at all. And when they pick up what I'm saying, they're like, oh, well, da-da-da. Now they've sort of learned something very different from what I was saying. And that is the same kind of messiness that I'm describing in the genome itself. It's sort of embracing the messiness.
And that's a feature, not a bug.
Exactly. And in the same way, when you're thinking about sort of these deep learning systems that will allow us to sort of be more creative, perhaps, or learn better approximations of these complex functions, again, tuned to the universe that we inhabit, you have to embrace the breaking. You have to embrace the, how do we get out of these local optima? And a lot of the design paradigms that have made deep learning so successful are ways to get away from that, ways to get better training by sort of sending long range messages, these LSTM models and the, sort of feed forward loops that, sort of jump through layers of a convolutional neural network. All of these things are basically ways to push you out of these local maxima. And that's sort of what evolution does. That's what language does. That's what conversation and brainstorming does. That's what our brain does. So, this design paradigm is something that's pervasive and yet not taught in schools, not taught in engineering schools, where everything's minutely modularized to make sure that we never deviate from, whatever signal we're trying to emit, as opposed to let all hell breaks loose, because that's the path to paradise.
The path to paradise. Yeah, I mean, it's difficult to know how to teach that and what to do with it. I mean, it's difficult to know how to build up the scientific method around messiness.
It's not all messiness. We need some cleanness. And going back to the example with Mars, that's probably the place where I want to sort of moderate error as much as possible and sort of control the environment as much as possible. But if you're trying to repopulate Mars, well, maybe messiness is a good thing then.